pesertanya udah masuk ya? Should we start now?
Yeah, we
Okay.
can start now, Pak.
Oke. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang dan salam sejahtera buat kita semua. Very good morning for our distinguished guest, Dr. RuAngelie Edrada-Ebel, who will give her talk about metabolomics. Before we start to our formal event, I would like to invite our Dean to deliver his welcome remark to our distinguished speaker. So please, Pak Dede.
Okay. Thank you, Mr. Yasman. Thank you. Welcome to Dr. RuAngelie Edrada-Ebel. Okay. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and good morning in the UK. Yeah, good afternoon to all my faculty leaders of MIPA UI, eminent speaker Dr. RuAngelie Edrada-Ebel, Dr.rer.nat Yasman, as moderator, colleagues and students of MIPA UI, ladies and gentlemen. Yeah, this afternoon, in such a privileged and honorable moment, to offer my sincere appreciation to our distinguished speaker, Dr. RuAngelie Edrada-Ebel from Strathclyde Institute of Pharmacy and Biomedical Science, who is willing to accept our invitation. Dr. RuAngelie, we share her inspiring talk with us with the title using Concatenated Analytical Data of Metabolomic Guided Production and Targeted Isolation of Bioactive Natural Product. Yeah. The utilization of metabolomic technique is to study secondary metabolic compound from organisms that have interesting bioactivity potential for application in the field of health, agriculture, or food. One of them and is effort toward drug discovery, so that with the technique identification, the targeted and untargeted metabolite profile can be faster, more effective. And Dr. RuAngelie track record of publication, mainly in the pharmaceutical sector, is appropriate for providing scientific update regarding this matter. In this MIPAtalk series activity is a firm commitment from my faculty to increase scientific insight in various disciplines, especially such as mathematics, physics, chemistry, biology, geography, and geoscience. This activity will be held every 2 weeks with the latest topic in various disciplines. We hope that from this lecture, fellow lecturer or student who are present here can get the maximum benefit and maybe open up the opportunity for further collaboration. Finally, we thank all for coming. We thank Dr. RuAngelie Edrada-Ebel for her willingness to fill this activity. Thank you very much. Wassalamualaikum warahmatullahi wabarakatuh. Thank you.
Thank you very much, Pak Dede. And we will have a photo group session first. So everybody who joined this Zoom, please on cam so that we can take a picture. Mbak April dengan Mbak Yusin bisa ya?
We are still waiting for some participants to open their camera, Pak.
Everybody who joined this Zoom, please on cam.
Okay, yeah, 2 slides. I will count 2, 3. For the first slide, 1, 2, 3. And the second slide, 1, 2, 3. Okay, thank you.
Okay, thank you very much.
Thank you.
So I would like to introduce you the rundown of this event. First, I will introduce to you the speaker today, a very short curriculum vitae of Dr. Ruan, and then I would provide maximal 1 hour for Ruan to deliver his or her lecture. And after that, we will continue to the session of question and answer. And then hope in next 2 hours we will finish this event. And I will try to first share the curriculum vitae of our speaker today. Oh, thank you, Yasman. So Dr. RuAngelie Edrada-Ebel, now working at Strathclyde Institute of Pharmacy and Biomedical Science. As we can see here, she is very prominent scientist with age index of Scopus of 45, education in Philippines for bachelor and master, and then she got his doctoral degree in pharmaceutical biology, Julius Maximilians University of Würzburg in Germany. She was as a senior researcher and instructor in Institute of Pharmaceutical Biology and Biotechnology in Heinrich Heine University Düsseldorf. And then she moved to NMR and MS Facility Manager in Strathclyde Institute of Pharmacy and Biomedical Science. Now she is an associate professor in pharmaceutical analysis, Strathclyde Institute of Pharmacy and Biomedical Sciences. Some currents of her publication is in 2022, metabolomic profiling of Malaysian and New Zealand honey using concatenated NMR and HRMS dataset, which I know will be one of his sample project for her talk today. Without further ado, I will let Dr. Ruan to start her lecture. So Ruan, time is yours now.
Okay, thank you very much, Professor Dede and Professor Yasman for the introduction. And I also would like to take this opportunity to thank you for giving me this session to introduce our research at the University of Strathclyde. So good afternoon, everyone. So I'll start to share my screen. See. Okay. Right. So yes, so for this afternoon in Indonesia, okay, I will be talking about secondary metabolomics. So what is actually metabolomics? So that is the study of metabolites after the end of a certain biological process. But then we are not really studying primary metabolites. So we are looking into secondary metabolomics because this is what we're interested on looking into. So this is mainly our natural products. So we're using secondary metabolomics to be able to target bioactive metabolites, to optimize harvesting of these natural products, okay, if we're looking into plants. And because we are also working a lot with microorganisms, then we also use secondary metabolomics to be able to see when these bioactive metabolites are being produced. So then the expected result will be to have higher yields of this bioactive metabolites. In this lecture, I would also like to introduce using concatenated NMR and MS analytical data. What does that mean? So we are just simply fusing two data sets to be able to have a significant result of targeting these different types of metabolites. Okay. My screen is not moving. There you are. Okay, just to give you a brief introduction of our group. So we are part of a multidisciplinary consortium. So we work along several countries in Europe. So sampling, for example, we do our sampling together with countries in Iceland, up here, and also southern part of Europe. We've got here the Mediterranean group, which is Greece. So we both get our samples in the colder regions of, which is almost near to the Arctic, as well as to the warmer regions of Europe. Bioactivity testing, we do a lot of collaboration with industrial groups. So one of them is in Norway, that's Pharmac. So they are actually working on agrochemicals. And then we also work with an Italian group, Axim, where they do a lot of anti-cancer bioactivity. Although in our group, at Strathclyde, we also have a very good group who's working with us on antiparasitics as well as anti-cancer compounds. So because we are multidisciplinary, we also work with genomics, and that is very important if you're doing optimization work. And we do have a very good group in Würzburg, where we've been working with them for the past 10 years. And then further work with industry within Scotland. We've got this industrial processing group, and we've got, for example, Ingenza, and then we've got the Scottish Association for Marine Sciences, and that is in the west coast of Scotland. And when it comes to seaweeds, we do work with a small medium enterprise that is the marine biopolymers. Then at the end, we need to do genetic and metabolomic engineering of these organisms, and we further work, for example, with companies which is a bigger company like Ingenza again, as well as with Heriot-Watt. So this is another university in Scotland. So we also do a lot of international collaborations, so both at every continent. So down here at Australia, we have a very good collaboration with Griffith University. There are several universities in Malaysia as well. Yeah, we've got Indonesia. But for today, I'm going to be presenting results with collaborative work with Malaysia. So we've got as well Egypt, Jordan, and in Africa, we've got Nigeria. Sorry, Egypt is here, and Nigeria. Okay, and our partners in Europe. So we've got a very new collaboration with France, and then we've got again Norway, and then going to the America. So what I'm going to be presenting is a collaborative work with Brazil. Okay, and we also have good collaboration with Mexico. So what is the challenge when working with natural products? So I always present this 4 quadrants. So the main challenge for everyone is, I think we all want to get new compounds, new bioactivity, and therefore we get into 100% novelty. So then we end up with a very good publication. The second quadrant is we get known compounds, but we've got new bioactivity and 50% novelty. So that's not so bad as well, because if you will see the drugs that we're using today, even for COVID, there's a lot of repurposing that's happening. So for example, the use of steroidal drugs from COVID, they were actually used before for, I think, as anti-asthma drugs. Yeah, but then they are being repurposed. So this one will also give us a very good publication in high impact factor journals. The one we don't want really is new compounds, no bioactivity, that's still 50% novelty. Well, we do get to publish these type of research, yeah, but maybe at lower impact factor, because as you would see, higher impact factor journals, they're always looking for new compounds with bioactivity. So if there's no bioactivity, but you have very interesting type of compounds, okay, then you still do get to publish them, but maybe at a lower impact factor. The big no-no in our type of projects is we get known compounds, known bioactivity, and known novelty. And this is the main reason why we went into metabolomics guided research work, because we don't want to land between these two squares or this fourth quadrant. So why natural products metabolomics? So what's happening here is actually basically we are embedding macros with natural product databases to be able to firstly enable rapid replication. Okay, and the main thing here is to use statistical or multivariate analysis software to investigate patterns in our data. And that is how we make quick decisions whether we target this type of metabolites. So here are 4 objectives. So the first objective, we use it as a selection tool. We could either use different plants or microbial strain, but the main purpose is to determine chemical novelty. Second, we also use it as a selection tool to optimize our process condition, which means we could integrate metabolomics data with the process variable data. So one example is linking chemistry with fermentation. But for today's lecture, one of my example is to link chemistry with harvesting time, which means we are looking at the different ecological conditions to be able to see when is the optimum time that the bioactive natural products are being produced. A very important tool is to investigate bioactivity, and you would see this in all the examples that I will be giving today. So this is to compare chemical profiles of bioactive and inactive extracts. So which means with these two, we are able to directly target our target the isolation of the bioactive metabolites in the first fractionation stage. And this is a very collaborative objective to integrate the results with genomic data, which means we're able to confirm the expression of biosynthetic gene clusters and investigate the biosynthetic pathway. So this is very important. For example, if you want to increase the yield of these low-yielding bioactive natural products. And this usually happens, for example, with microbial metabolites, where you sometimes would get less than microgram levels of these new natural products. So this is our typical metabolomics workflow. We start with mass spectrometry. We use switch mode, and I'll tell you later on why that is important. So switch mode, we use both positive and negative ionization mode. Once we have the data from our mass spectrometry, we do our data processing, and we use the platform MZMine, but there are also other freeware platforms available. So I think there's like XZMass, and that's also available on the web. But we use MZMine because that is that was put together by Finland, where we have a very close collaboration with. Okay, so here we do picking, convolution, the isotoping, chromatography alignment, and that's very important because if you have 100 samples to analyze, you would imagine, or you would be able to observe that some of your peaks are starting to migrate. Yeah, and that has something to do with the stability of the column. So chromatographic alignment is very important. And lastly, formula prediction. And this is a prerequisite if you want to do the replication studies. So once we've predicted the different formulas, then it goes into our database. So we have our match. So we try to match the different predicted formula, whatever we find in our databases. So we use commercial databases like the Dictionary of Natural Products, but we also have like internal databases, which means we have a collection of compounds that we have isolated in the past, and then we match it with the molecular weight and the retention time. Okay. And then finally, we do multivariate analysis. And in this part, we already integrate the results of NMR and bioassay. So this is the part where we start to define our target metabolites for further isolation work. And once we've isolated them, we do the structure search and then do further structure elucidation. So we use this Metabolomics workflow in all types of our project. There will be maybe some tweaks depending on the difficulty of the project we are handling. So just to give you a brief introduction again on our analytical platform, we use both mass spectrometry and NMR spectroscopy. But if you look at the literature, they also use other platforms. It could be a combination of mass spectrometry and infrared, or you've got then UV and NMR spectroscopy. So they have all these different methods. But for us, mass MS and NMR works very well. So basically what we're doing here is we're trying to use the advantages of both methods. So we know for mass spectrometry, we're able to see lower concentration of metabolites. We could even go down to picogram level. The only disadvantage of mass spectrometry is quite critical where these compounds are ionizable. So some of them will be ionizable in the positive and some in the negative mode. The best example I could give you are the phenolics. So phenolics, they will not ionize in the positive mode. They only ionize in the negative mode. And that's the reason why we use switch modes. Another thing is that polarity also plays a very important role in mass spectrometry, because usually we're using polar liquid chromatography, which means we have difficulty in visualizing nonpolar compounds, which means it's better to use other methods of MS. You know that there are a lot of methods. We've got electron impact, we've got soft ionization. So we need to use a combination of those as well to be able to see everything. However, when it comes to NMR spectroscopy, the great advantage is we are able to see a good picture of everything that we are able to isolate. So which means NMR is not dependent on the capability of the compounds to ionize. So you have a good picture saying, okay, these are the compounds that I expect, and we are able to isolate. The disadvantage of NMR is that we are not able to detect compounds down at nanogram levels. So the only metabolites that you could detect by NMR are those at greater than or at microgram level. However, as I've said, this is the level of compounds or metabolites that we are able or feasible to isolate. Okay, and when it comes to preparation of compounds or samples, it's easier to prepare samples for NMR rather than preparing samples for mass spectrometry. Okay, so these are the three application projects that I would like to introduce. So the first one is antitrypanosomal compounds from an endophytic fungi that was isolated in the coastline of Malaysia. And this introduction project here is just to show you or illustrate to you how we use a metabolomic guided isolation work rather than doing a bioassay guided. So I'll show you the advantages and disadvantages of metabolomics in this first project. The second project is assessing the seasonality of Brisonema intermedia from the Brazilian Cerrado flora. So this is already a concatenated method of finding biomarkers of certain bioactive or anti-inflammatory compounds from a certain plant, which is Brisonema in this case. And the last project I would like to show you is comparing Malaysian and New Zealand honey. So this is quite an interesting project, and you'll know why later on when we get into that. Okay, let's go to sample project 1. So this was done by a Malaysian student of mine, and RuAngelie Yasman, who's working at the University of Malaysia, Terengganu. So we collected the mangrove at this coastline of Terengganu. It's a very nice coastline. Okay, they are actually famous for their turtles. Okay, so what we did was to collect parts of the roots and the twigs, and she was able to isolate 7 different fungus, but 3 of them were only active. So we've got Fusarium, Lacidiplodia theobroma, and Aspergillus aculeatus. But for today, I will just be presenting her results on Fusarium. So after inoculation and isolation, she was able to identify the different fungi using molecular work. She optimized them using 2 types of culture, that's the rice and the liquid media, and at different incubation periods, 7, 15, and 30 days in a 500 ml flask. And then she did an extraction, and then did the replication studies, as well as metabolomic analysis, together with NMR, as well as her bioassay results. And finally, she went into the scale-up by choosing the best media and the best incubation period. And further on to bioassay screening, but this one is again together with metabolomic guided screening or isolation work. And finally, she has her pure compounds, and then she further elucidated the structures. So this is the result of her culture optimization work. So as I've said, she had rice culture and the liquid culture. So incubation period was 7, 15, and 30 days. As you would see here, the results were actually, she always had 99% inhibition of trypanosoma in almost all of her cultures, except for the rice culture at 7 days. However, maximum yield was attained between the 15 and the 30 days rice culture. So the question is, which one will be the best? So looking at the chemical profile, so this is from the high resolution mass spectrometry result, we could see that the 15 days has more blue lines, and they are more diverse from polar to non-polar in comparison to the 30 days culture. So in the 30 days culture, there will be lower molecular weight compounds. Yeah. And here, when you go to the higher molecular weight compounds, nothing is really produced. So we could consider that the 15 days culture is more diverse. And that's the reason why for the scale up, RuAngelie chose to work with the 15 days culture because of its more diverse chemical profile. This was also a win-win situation for her, because then rather than waiting for 30 days, 15 days, you do your isolation or extraction work. So which means it's also quicker. So if you translate this in an industrial point of view, I think the industry would rather work with a shorter time of fermentation period rather than waiting for a longer 1 month period of incubation or fermentation time. So let's look at her results. So remember what I said about the switch mode? So here is the total ion chromatogram. The red line represents the negative mode, and the blue line represents the positive mode. As you will see here on the total ion chromatogram, we've got more peaks that we could visualize on the negative mode, while there are almost only 1 or 2 peaks that we could find in the positive mode. So that is the great advantage of working with both positive and negative mode. I do know some laboratories would just be routinely working on the positive mode. So I think you'll be missing a lot if you're not working on the negative ionization mode. So here is a scores plot, just to give you a brief introduction. So what you see here, each of the circles there represents the different fractions. Okay, so on the left, we've got the active fractions, and on the right is the inactive. So that's the so-called scores plot. So we could also translate that to a loadings plot, the one you see here on plot C. And the way to interpret this, these individual spots you see here represent the same fractions that appear on the same quadrant on the scores plot. So which means, for example, this fraction 10 and 12, you have this metabolites with molecular weight 331, 659, and 329. So which means we could define this, or Winnie could define this as her targets. Looking at fractions 4 and 8, then these are the responsible metabolites for their bioactivity. So you have here one at 295 molecular weight. Yeah. So this is actually negative mode, which means the actual molecular weight will be 296. So what you see here are all M minus H. Okay, so 659, so which means that's 670, 329, so the molecular weight will be 330, and 331, the molecular weight will be 332. Okay, so those are the target metabolites that she needs to isolate. Now, just to validate this result, she did another plot. So we call this S plot. It's just plotting the active metabolites versus the inactive metabolites. Okay, and these are the target metabolites. Now, the take-home message on this slide is that if you compare these results with the chromatogram, okay, if you are not working with statistical data, the first attempt of a student or any researcher will be to isolate this intense peak. However, as you would know from the multivariate analysis, these compounds are actually inactive. So when you test the pure compound, you will end up with inactive metabolites, which is quite unsatisfactory because then you're just wasting a lot of time and resources. So if you look at the relative abundance of these compounds, they are just down here. So they are these small components here. So which means Winnie could just discard these metabolites and work or focus on this target metabolites. So that saves her a lot of time and effort as well. So prior to isolation work and structural elucidation, she was very happy with her results because as you would see here, the IC50 is very much comparable to the known antitrypanosomal drug Suramine. Yeah, so Suramine has an IC50 of 0.071. Solanol is even better, 0.047, and Javanicine, okay, compound 2, is very much comparable to that of Suramine. And the best thing is if we look at the selective index, yeah, we know the problem with Suramine is quite toxic, so which means it does affect normal cells. So, but when we look at the selective index, and Javanicine has 915, and Solanol, which is even more active with more active than Suramine, is less toxic than Suramine. So it has an SI index of 2769. So that's a very high selective index, yeah, which means Solanol at the end is a better drug to be developed than Suramine. So now let's go to the next project, where the next two projects where we start to fuse the data sets. So you don't need to be confused with this slide, because I know you're seeing here a lot of mathematical and statistical formula, but basically what I want to show you here is we just simply put together the mass spectrometry data, as you would see here on the upper part of this table, and down here is the NMR data. So we fuse this, okay, and we get the sum of the standard deviations of each of the respective blocks. So there's a lot of papers already out there to describe these type of methods and how to do this in Excel. So it's really very simple, because then Excel already has this automatically. So you just need to call back the formula and then you're able to get it down to work for you. Another way to do it as well is to do it with statistical softwares. We're using SIMCA, but I think there are also other statistical softwares available there that's free on the web. And you also have this this possibility to do such fusion of data. So what we're using here, as I've said, is mass spectrometry and NMR, but down out there in the literature, you can see also combinations of UV and IR, UV and MS, okay? So you could just play around with these different data sets and see what works best for you. Okay, let's go to the second sample project where we use fused data sets. So here is a project, we were trying to find out the biomarkers in Brasonima intermedia from Brazilian Cerrado. Okay, so just a background of the project. The first thing is the local population of the Brazilian savanna uses the bark of Brasonima as infusion because they were found to possess antiseptic, antimicrobial, anti-hemorrhagic, and anti-inflammatory properties. However, the main problem with this project is that the impact, if you inspect MS and NMR spectral data visually, we really cannot find clear differences to indicate the different biomarkers at different seasons. So Anna's hypothesis is that seasonality plays a very important role. It's a major environmental factors that could exert influence on the synthesis and accumulation of secondary metabolites in Brasonima, particularly triterpenes. So that was her main aim. So her objective is to target seasonal biomarkers from unique perceptible constituents, which were really low yielding, particularly the presence of anti-inflammatory triterpenes during the summer. So she was comparing these different samples from different seasons, and she was not finding these differences. So we said, okay, why don't we use a metabolomics approach to make this significant and make this more obvious for us to say, you have only to go to the Cerrados once a year, and this is during the summertime. Okay, so these were the results. So first one here is just mass spectrometry. So it's not fused, it's just pure mass spectral data. And the problem we could see here is that there's our overlapping of samples between the spring and the autumn. We do get a very good separation, but relatively good separation of the summer. Yeah, but still, you still have one overlap for one of the replicates. When we come to the NMR, this is quite confusing because this time we've got an overlapping of the samples between the summer and the autumn. So which means that would tell us that the samples were quite really similar in this case. But when she concatenated, she fused the data sets between mass spectrometry and NMR, we could see a trend and a good separation of the summer samples. So there's a sloping trend of the production of metabolites between the winter to the spring, and then we get a good separation of the summer, where she was hypothesizing that we get the highest yield of the anti-inflammatory triterpenes during the summer period. So she separated the summer from the rest of the other samples, okay? And here from the NMR data, she could see then the signals that represent the triterpene compounds, and those were the chemical shifts between 1 to 5 ppm. Looking at the mass spectral data, so these are all fused data. She was just extracting the data after fusion. Looking at the mass spectrometry data, she was also getting the right molecular weight of the expected triterpenes, okay, which was between 300 to 500. To validate her work, okay, she used this whisker bar graph. So the purpose of this experiment was to find out the relative abundance of these individual triterpenes that she was able to find from the loadings plot. So as you will see here, so these are the 3 unique triterpenes that was collected from the summer samples, okay? And you can see a very good relative abundance of these oleanine, and lupain triterpenes. So this is N2, N23, and P4128. However, we were also able to see when the flavonoids are being produced, and they were indicators of spring samples. Yeah, so you can see here a large whisker bar graph, okay, for the triglycosylated flavonoids and glycosyl flavonoids, which were only occurring during the spring season. So, and finally, to conclude her work, okay, so what are the requirements for increasing the yield of triterpene production during the summer? So these are the requirements. We need a higher temperature. It should be greater than 23 degrees centigrade. Okay, solar radiation should be 2000 to 2500. Lower humidity, so percent humidity should be less than 70%. And rainfall should only be between 0.10 to 0.15 millimeters. So let's go to the last project. This is comparing Malaysian and New Zealand honey, which was quite controversial because Yasman was able to find out that the Malaysian honey was bioactive against the breast cancer cell line, while the New Zealand honey was inactive. So the main aim of her project is to use honey as a prophylaxis against cancer. Okay, but when we look at and inspect, or if we inspect the MS and NMR spectral data, it doesn't really indicate any differences between the major components. So we could only see these sugar components like glucose, fructose. So those are just the main components that we could see, and they're very similar between the New Zealand and the Malaysian honey. So the question is, yeah, what is the real difference that make Malaysian honey active and New Zealand honey inactive? So her hypothesis is to fuse the data set that will improve the clustering and correct the differences between the replicates and define the bioactive biomarkers and differences between these two geographical type of samples. So, and finally, she will be able to pinpoint the low-yielding bioactive biomarkers from the more perceptible sugar constituents. So let's look at her results. So this was before fusion. You can see the NMR data. On the left are the Malaysian samples, and on the right are the New Zealand samples. But we could see here in the middle that there is an overlap between the Malaysian and the New Zealand samples. If you look at the loadings plot, okay, so we said on the right is the New Zealand samples. So we could see a higher concentration of chemical shifts between 4 to 2 ppm. Yeah, so those will be, again, sugar components and lipid components. But if we could look at the Malaysian samples, you have here a higher concentration of the greens and the oranges. So that's 5 to 7 ppm. So that represents chemical shifts that are aromatics, phenolics, yeah, particularly between 5 to 6 ppm, and they could also contain some sugars, so 3 to 4 ppm. So you will be thinking in the directions of glycosylated flavonoids. Looking at the mass spectral data, we've got better separation between Malaysian and New Zealand samples. Here you've got darker blue, which is for lower molecular weights for the New Zealand samples. And on the left, you have lighter blue dots, and those are for higher molecular weight. So it could be between 200 to 400. And that is the Malaysian samples. While going back to the New Zealand sample, that will be between 100 to 280. Yeah, but the difference is still not that obvious. I mean, the difference in molecular weights are quite close, and we consider that as non-significant. So we fuse the data set, we get better separation between the Malaysian and the New Zealand samples, but surprisingly, we've got two clusters between the New Zealand samples, and this one are from the north, and this one is the south. So the question is, what is happening here? But first, let's make our first conclusion between the differences between New Zealand and Malaysian. Yeah, we could also see that, or validate that, we have a very good result here, because from the HCA, or hierarchical analysis, we could see a very good separation between the Malaysian and the New Zealand sample. So the dendrogram could separate those two types of sample. So this is the conclusion from this set after fission. So the New Zealand honey has higher lipid content, and this is represented by the higher density, you can see here, of darker blue dots. Okay, so that is in terms of their chemical shifts in NMR spectroscopy. Looking at the lighter green dots, so that would represent the lower molecular weight compounds. So that data is from the mass spectrum. Now, looking at the Malaysian honey, there was a higher sugar and polyphenolic content, and that is represented by the higher density of lighter blue dots. Okay, so that is again the NMR data result. While there is a higher density of yellow greens to orange spots indicated higher molecular weight compounds. So that already validates our hypothesis that the Malaysian honey had higher content of glycosylated flavonoid compounds, while the New Zealand honey is more like lipophilic, containing a lot higher amount of fatty acids. So looking back on the separation between the North and the South, yeah, so here on the left, we've got the South New Zealand honey and the North New Zealand honey. So what is the main difference here? So again, we have here our S plot. So these are the individual metabolites that represents the North and the South, yeah. And we could see here, if we extract these data points and put them into a bar graph, so let's look at the South first. So from the South, we have a molecular weight ranging at between, so around 300, so 300 to 400, yeah? And there is a good expression of the presence of phenolic compounds with chemical shifts between 5 to 6 ppm. However, the northern samples, okay, you have here lower molecular weight compounds, so it's only between 150 to 300, and there is a larger concentration of acetylated compounds. Yeah, so those chemical shifts to 2.5, 3 ppm, so those are acetylated groups, and also a higher sugar content in that case. Okay, so that explains the two clustering that occurred between the northern and the southern New Zealand. And if you look at the map of New Zealand and their geological conditions, the northern island and the southern island are also very different. So one is more temperate and the other is more tropical. And maybe that also explains some of the overlapping of the samples between New Zealand and the Malaysian honey. However, looking back on the Tualang honey from Malaysia, we could also see that the type of chemical profile is also very different than those found in New Zealand. And these compounds are very typical of the type of plants that you find in Malaysia. So we've got here Curcuma longa, Plumera rubra, Cocos nucifera, and those are plants that you don't find in New Zealand. And according to the dereplication, this could be the bioactive metabolites responsible for the anti-cancer bioactivity, okay? And these were not found to be present in the New Zealand honey. So just to summarize and conclude, so with fused data sets, we were able to validate and optimize the production and harvest method for the target metabolites. Fused data also improved the linearity of the production and the presence of the metabolites against the respective parameters considered in these studies. And finally, the fused data resulted to better clustering and corrected the difference between replicates to define low-yielding biomarkers. So which means we could discard all these major components, yeah, and look at these minor components, which really defines the significant differences between samples. This could be different in fermentation conditions, differences in geographical environment or seasonality, which as I've shown with the Brazilian project. And finally, I think I have illustrated or demonstrated that metabolomic tools did provide us a clear and well signposted map to our target destination. As we know, natural products research could be like a maze. Yeah, you really need to go through all this to be able to go to your finish line. But then I think metabolomics give better signposts. Yeah, and even better than our road and traffic signs. I just have some examples here. So these are real examples, like you've got this roundabouts within roundabouts, and this is very common in Southern England, or this is somewhere, I think, in the States. So which arrow do you follow? We don't want natural product research to be like this. Keep right, but then the arrow is pointing left. Or we don't want to see any beware signs of any road surprises. So that is very important when we're doing our isolation work. Remember, I told you in the beginning, if we tend to focus our attention on the higher peaks, then what will be the surprise? We get an inactive metabolite. Yeah, but then with metabolomics, we're able to focus on the right target metabolites, and those are usually the peaks that are considered to be minor on our chromatograms. And of course, we don't want to waste any of our resources and efforts, and we don't want to have this sign in front of us wrong way and go back. So finally, I would like to thank the people who work on this. So my postdoc, Dr. McIntyre and Dr. Tsang, who did all the standard operating procedures when it comes to metabolomics analysis, the work of Dr. Maslan and Dr. Yusuf from Malaysia, and Dr. Sanata from Brazil. And of course, our funding agency. So Brazilians are funded by FAPEP, CNPq, and CAPES. Yeah, we've got also good funding from the European Union, which is quite a problem now after Brexit, because we do get a lot of good Erasmus students who support all these projects that we have. And finally, I would like to thank you for your kind attention. So now we could open the next session, questions and answers.
Thank you very much, Tuan, for a very nice presentation.
You're welcome.
Now we come to the question and answer session. If any of you have a question, just raise your hand and open your mic. Or as written by the operator, you can also type your question and I will read it for one.
Was I too quick?
Before we start the question and answer, let me just to say hello to Pak Haidir from BRIN, Budian from UNAN, Bu Triana from UGM, and what's next? Pak Edi, ya, Pak Edi from Polytechnic Bandung. So again, if
Dian is also there.
Yeah. Good afternoon, Budian. Thanks for joining this event. Any questions? Ruan.
Yeah, go ahead.
I know that one of your sample project dealing with the environmental metabolomic, the one that from Brazil.
Yes.
Is there any example, more example for environmental metabolomics in your group?
Yes, so all the work from Brazil is usually ecological. Yeah, so I think that's the second project. We had one more project, I think, where we first tried the concatenated method. That was also an ecological project with Brazil. Yeah. So most of our projects in Brazil are ecological. So, and yeah, the papers are there. So I think.
And during your project, instead of the interesting bioactivity, is there also a new compound? As you said that our target is actually as a good one is both new compound and also new bioactivity.
Yes, right. So for example, the triterpenoid compounds is
triterpene is a new compound from the
those
Malaysian.
are new compounds, right? Yeah. So usually what we do is we isolate first the compounds. Okay. Okay. So they are published in another paper, and then we test the optimization when to harvest them, and then that will be we use a different approach. So just like the way we did with the first presentation for the mangrove plants. So the approach is a little bit different. Yeah. So the metabolomics isolation approach is different when we go into the optimization approach. So we usually use the fused data sets when we want to optimize the production of these metabolites. So this could be fermentation or harvesting period. But the best thing is first isolate the compounds and find out, okay, these are the type of compounds, how do we optimize them? And if we do microbial work, so that could be fungal or bacteria, then other collaborations comes in as well, because then sometimes you just cannot optimize them by changing the media. You need to do some genomic elicitation work. Yes. So molecular biological works, and we need collaboration in this space. So there's a lot of involvement of other groups. Yeah. When we want to go into finding novel compounds.
Thank you. Again, is there any question from the audience?
Or anything that you need clarification?
Bu Sita and Bu
Busita
Astari. First goes to Bu Sita.
Yeah.
Thank you, Pak Yasman. Ruan, I was just trying to digest all the information you gave us. But a simple question then, since I'm also working in microbiology and trying to work on biosurfactant from hydrocarbon degrading bacteria. I am just wondering. I've never done what you did. I mean, your work is not that familiar with me, but your title says that it's for targeted isolation. Does that mean? And this is kind of a basic question. Does that mean using metabolomics will save us some in quotes pain in detecting these products, or what is there the really big advantage of that?
So it really is the great advantage is save time. It saves us a lot of time and effort to isolate everything. Yeah. So when I was doing my PhD, long time ago, I isolate everything. Doesn't matter whether it's active or inactive. Yeah. But now, especially in the UK, a student is only given 2 years to do their lab work for their PhD and 1 year write-up. So unlike in, I think, Germany, we're given 4 to 5 years, right? And that too, if you're given 4 years, then you work 3 years in the lab. But if you're given 2 years, that's really very quick. So which means, I think with this method of targeted isolation work, then they know what their target is in the first fractionation, and they just target that. And that will be more open to faster or quicker publication as well, because we're 100% sure that what we are targeting is bioactive, okay, and non-inactive as well. And another thing is, for example, I think when it comes to structure activity relationship, okay, you could always go back to these plots, yeah, and find out, okay, do I have some structure that's related between my active and inactive? Then you could go back in that plot and then start isolating them and build up your structure activity relationship. So it has been really faster. So I think one of my postdoc, when she started this project, so she was targeting 12 compounds. So this is anti-TB. I cannot show her results because it's collaboration with the industry. She only did the entire project for a month, maybe because she's a postdoc. But she said it's just because the project, you don't need to look at, for example, you've got 22 fractions to look at, and you only find the activity in 3 fractions. Then why do you need to work for 22 fractions? You just focus your attention into 3 fractions, right? Yeah. So that saves us a lot of time and effort. Yeah.
Oh, so thanks. Very enlightening. I mean, of course, the initial investment for doing this is quite high, but yeah, I can see in the future that the approach will be different for the younger generation.
Yeah.
Okay, so yeah. Food for thought. Food for thought for our dean for the investments on the equipment.
Yeah, right. Yeah, that's true. Yeah, the investments could be high, but then the daily consumables or daily operating procedures becomes cheaper at the end. Yeah, because you just tell a student, I don't want those inactive compounds, especially if you're working with the industry. The industry doesn't care whatever interesting structure you isolated. If it's inactive, they throw it out the bin, right? They just want active compounds.
I mean, the days of the large screening with thousands of plates are probably disappearing right now, I think.
Yes, and I think the industries has also become more focused. Okay. We create all these libraries.
All right.
But then, what the industry at the end wants is just, we just want something active, something that works for us. Yeah.
Thank you.
Okay, thank you, Bu Sita. Ruan, I will read the question from Bu Ari. Hello, Bu Ari. I'm sorry I missed to mention your name. There are 3 questions from Dr. Arianti. First, what is the minimum number of samples to be statistically accepted for this type of experience? Should I read all the question, or I read it one by one?
Maybe one by one, because I'll forget.
First, what is the minimum number of samples to be statistically accepted for this type of experiment?
Okay, so you can have 10 samples, but the most important thing is replicates. Yeah, if you don't have any replicates, you could forget everything. So it'll be 10 times 3, you've got 30. Okay, so 10 samples. So for example, if you're comparing different fermentation conditions, different season, or I think Anna had 4 samples because 4 season, but then she had 4 replicates for each, or I think 5. So that will be 4 times 5, 20 samples. The replicates is the most important thing. You could also
the minimum replicate is 3.
The minimum replicate is 3.
So we can know the deviation standard of the data.
Right. It's best to have biological replicates.
Okay. Then the second question is, were the sample subjected to the NMR and MS in the form of crude extract or fraction? or even the pure compound?
Okay, for the initial screening, they were all extracts.
Okay.
Yeah, so for mass spectrometry, just to give you
From
some
that, then you targeted the compound that we are interested in.
Yes, so we start with crude extracts. So both we subject them both with MS and NMR just to find out, okay, which is the best extract to work with. Once we've done that, we take the extract, we do the scale up, and then we do the fractionation. So fractions, usually initial fractionation, you get maybe 12 to 20 fractions. Okay, and we do again MS and NMR of that. And we do a metabolomics approach, and that is the first time we target the metabolites. And once we've targeted the metabolites, we do not do any bioassay in between during the entire isolation work. So then the next bioassay will be done on the pure compounds. So without doing any bioassay work in between isolation, that saves us a lot of samples. Yeah, just imagine if you do your bioassay every step, then you'll be losing at the end.
Yeah,
You will end up with less than a milligram. You cannot elucidate your compound anymore. Yeah.
Okay, last but not least questions. How to deal with multiple dark ions that might complicate it to the MS data?
All right. Oh, that's okay. We have this MZ-Mine, right? I've mentioned that. We love MZ-Mine because it does tell us which one is the multiple ADOC. So you just so you process your data, you put all your spectral data into this MZ-Mine platform, and then it will tell you, for example, which ion peaks are together. Yeah, so for example, you have an ion peak at 150, 300, 600, 1200. So MZ-Mine, what MZ-Mine will do will say 300, it belongs to 150. 600, it will again tell you it belongs to 150. It will also give you the retention time. So you know all those multiple atoms belongs together. When it comes, for example, to, you mentioned adduct, like the presence of sodium ion, magnesium, potassium, it will also tell you. Yeah, you just need to identify these parameters to the platform. Say, okay, I'm expecting natrium adduct and potassium adduct. Because this type of adduct is also dependent on the type of instrument you're using. So for example, Orbitrap, we don't expect any natrium adduct, but I think when we use Agilent, there's a lot of natrium and potassium ion adduct. So we just need to put that into our parameters when we're using the platform.
The MZ-Mine program is open source program or it is a licensed program?
It's open source,
so
everyone could use it.
Oh,
yes,
really?
Yeah,
Nice.
And yeah, we've prepared already. So if you look at our publications, you could just follow our parameters as your starting material, as your starting parameter. Another thing is that if you go to the website of Scripps in San Diego, they also have starting parameters for certain projects. So if, for example, you're working with GCMS, I think yeah, the Scripps Institute in San Diego, they have a very good protocol on GCMS analysis. So it doesn't need to be LCMS or it doesn't need to be an Orbitrap. But if you want to work on Orbitraps, then you could look at our publication. We have the parameters there, and you just could follow our parameters just as your starting point. Yeah, it is always nice to have a starting point.
Okay, thank you very much, Ruan. Before I go to Bu Dian, I would like to pass the chance to Bu Astari. Bu Astari?
Yeah, okay. Thank you, Pak Yasman. Thank you, Dr. RuAng, for the interesting talk about this fused data in metabolomics. My background is not metabolomics, so I learned from Pak Yasman.
Thank you, Astari.
So the first one is related with the, you show us the research comparing 7 days and 15 days and 30 days in the beginning of your talk. And you said that the 15 days is the most optimum one due to the more diverse chemical compound. So my question is that is it always the more diverse one is the better one? For example, if we have the targeted chemical, which is with the lower chemical molecular weight, and then because in your data, the 30 days yield is better. So is it depends on the aim of our study, or the more diverse one is always better?
Well, it depends actually on the aim of your study, yeah. But here, if you look at the 15 days and 30 days, the yield is not really that significant difference, yeah. So one was 900, I think 979, and the other one was 103. So if you round them off, they're both at around 100 milligram, yeah. So we could discard that as our as a parameter in choosing the between the 15 days and 30 days. And then the second aim is to look at the diversity. And Nurwini was actually telling me that she will get more compounds in terms of diversity from the 15 days rather than the 30 days.
Okay.
Yeah. So,
then I'm interested in the comparison between Malaysian and New Zealand honey for the cancer agent. So my question is, due to your explanation, the Malaysian honey contain more glycosylated flavonoid, right, compared to the New Zealand one, and also you compare the north and the south one. My question is, which factor has more effect on the chemical composition, whether it the geographical condition, or for example the variety of the bees, or even the host plant?
I think it will be the plant resources, because then the bees take the
from the plant.
Yeah. Yasman knows this because I mean his project before, so the snails were foraging on the sponge. So it really depends what were.
So it's mean biodiversity is very important.
Yeah, the environment is very important. Yeah, so depends on the type of plants that they were foraging on. But we always knew it was controversial for us when we saw the results, because we know that New Zealand honey is usually manuka. Yeah, manuka is a kind of plant, and it's really widely distributed in New Zealand. And there's a lot of studies about manuka that it's really good as a prophylactic for cancer diseases. But then why in this result we weren't getting negative for New Zealand manuka, and we were getting better results for tualang, Malaysian honey? So yeah, and then we did just see that the chemical profile was entirely different as well. And then we look at the individual studies, because all these compounds are anyway known, so it's not, there's no we don't need extra effort and resources to re-isolate those compounds. So we just look back the literature and see what has been reported on these type of compounds, and we would see that they have been reported for antioxidant activity, for anti-cancer activity. And we also saw from the literature that manuka honey, although they have been widely used as prophylactic, that the viability of cells, of anti-cancer cells, of cancer cells, is only really down at 60%, which is very comparable to what we got. Yeah, the only difference in our study where we were able to define what type of metabolites we were finding in manuka and tualang honey, which have never been reported before. Yeah, so that became our advantage using the metabolomics approach. Yeah, but all our results were also what we call this parallel to what has already been reported. Yeah, we just showed more evidence. Okay.
Great. Okay, last question. Yeah, you conclude that there are a lot of advantages of this fused data in metabolomics. My question is, what is the challenges faced using this fused data in metabolomics?
Okay, right. Finding the right model. Yeah. So, well, I didn't go to this discussion, so because yeah, so finding the right statistical model, you have to, for example, play around with the scaling of your data sets. If you need to transform your data sets, that you don't lose any information. So if you look at the literature there, they do a lot of log transformation. But we also found out that if you do that, we will be losing a lot of information, okay? Because log transformation could average your data set, and we don't want to do that, yeah? Because we want to see still the actual data points, and we don't want an average of those data points. Because those data points will give us the significant metabolomic feature to define, for example, our aims and objective. Yeah, so examples of these aims and objectives is, for example, defining bioactivity, yeah, defining differences between these geographical honeys, yeah. Because I think one of our referees, when we submitted this paper, said, "Did you do log transformation?" I said, "Yes, we did, but we just get a straight line where you're losing." So I think that is the challenges when you're looking at statistical data set. You need to try a lot. There's a lot of models available there, and you could see that if you're using Simca. I think there's another program that you could try, which is a freeware. It's called Metaboanalyst. Yeah, you could play around the program. You could generate a lot of different type of models. Yeah, and you just need to find the right model that would answer your question. Yeah, but then in saying that, yeah, you need to also prevent of being biased. You just don't want to see good visualization. Yeah, you need to see whether, okay, am I discarding bad data that that are really important? Yeah, you don't want to do that. Yeah.
Okay. Okay. Thank you, Ruang.
Thank you, Bu
Pak
Astari.
Thank you, Astari.
Then we come to Budian. Silakan, Budian.
Oke, makasih Pak Yasman. Thank you very much Pak Yasman. Halo Ruan. It's nice to meet you again. Oh
You
my
too.
God. Oh yeah. So feel report about your success study, and now I want to tell to you that just in moment we starting to make a study OSMAC approach with our FUNJ, Ruan.
Yeah.
And then for that, we have to analyze the metabolite compound of the OSMAC and the standard media of FUNJ with LC-MS, right? And what is our problem is in Indonesia we have no library, complete library about the natural product special from FUNJ. So we find it with what you have call it MZ-MINE, right? To find the molecular weight, and then Rainer have mentioned that to find the molecule to find the substance, we have to open the GNPS. Can you tell me about the GNPS and how to access it? And that is the first question. And the second question, what is the requirement so that our study can submit to the good journal? What is the requirement? How deep is the data, the analysis, and so on?
Okay.
You get that? Okay, thank you, Ruan.
I'll try the end. Yeah, molecular networking, yeah, is a good data. They have a good library. So I think when we started working with molecular networking, yeah, we cannot really find anything because they were just limited to actinomycetes. But now they have increased their library, so you could definitely work with that. But you just need to be careful because it's not as what do you call this? It's not as intensive as Dictionary of Natural Product. But then when you work with DNP, you are working with just high resolution mass spectral data. There's no MSMS. And that's the great advantage of molecular networking, because you've got this MSMS data, which means you are able to network the similar compounds or derivatives together in molecular networking. The way to go do it is you just go to the Scripps website. If you just Google GNPS, it will get you directly into the website. I forgot the name of the program, but it will tell you there's a step-by-step procedure, which program you will need to be able to do molecular networking. Yeah, I think so. Yeah, I've never done it myself. Only my students are doing it.
Okay.
But yes, yeah. And then the other question, yeah, good journal. So the requirement if you're working with Metabolomics is that you also have to show your data. Yeah. So there's like this, because it helps them build the database as well. So for example, you could mention GNPS, and then you have to store your data set, your raw data set into GNPS. Or for example, Strathclyde, we have our own data set procedures. So then we just give the website of the website for Strathclyde in managing our data sets, and which means this is open to the public, and everyone could see how our raw data set would look like. Yeah. But personal point of view, I mean, okay, I could see everyone's data set, but what do I do with that? Right? So it's just a requirement of the journal. But
yeah,
well, I had one.
You are the one of the top reviewer of the top journal, Q1 and Q2 Scopus journal.
Yeah, I said as a requirement, yeah, you have to put your data set on the web. I mean, it should be open to the public.
Yeah, the novelty. If I saw only the LC-MS data and then match with the GNPS, is this enough for publication? I mean,
yeah,
or whether I have to find a new compound from the osmotic approach, so that we can submit it to the journal.
All right. So finding new compounds is getting more difficult. So that's the reason why we get into developing new methodologies. So maybe I would say one of the strengths of my group is we've been developing new methodologies.
Yeah, right.
And that's
the
the reason
is
why
the new developing for us.
Yeah. So
here we in Indonesia we have no new methodology, so we have to find the novelty new compound.
New compounds.
Yeah. Right. Yeah.
Yeah. Well, when you're trying to find new compounds, then you have to really have very good.
It is so difficult.
Bioactivity.
It is so difficult just now to find a new compound.
Yeah.
Only with if I use the isolation with column chromatography only. This is the big problem for us.
Yeah, then you could try molecular networking if it has never been done, for example, for Indonesian fungi. So that would be something, some good aspect of that. Yeah.
Okay.
Yeah.
Still in collaboration, right? Thank you very much.
Right. Thanks, Dian.
Yeah, I hope Pak Prof will glad that we have we always make a reunion. Makasih Pak Yasman. Assalamualaikum. Bye, Ruan.
Bye. Thanks, Dian.
Thank you, Ruan.
I'm sorry. Now we come to Djuhana.
Hello, thank you, Yasman. Hello, Ruan. So glad to meet you again.
You too.
Okay, I would like to ask you mentioned about using LCM, SMS, and also NMR for the metabolomics, right? You mentioned in UGM in my faculty, mostly we use FTIR to do the metabolomics. So in order to find the potential new compounds, which one that you find that's best, or is there any specific equipment for specific purpose or anything? Like for example, you mentioned about the different chemical content in honey. That's really interesting. in this case, that will be the purpose will be more to like authentication or like a quality control, or is there also possible to develop it as also for new compound finding like you mentioned previously? Thank you, Ruan.
Yeah, thank you, Triana. Yes, of course you could use IR as well. So I mentioned in the beginning that there are some groups who are using IR as a metabolomics approach. Yeah. The nice thing about IR is you could really define functional groups, but I think for IR you need to use a lot of standards if you're comparing compounds. So that will help you in the dereplication as well. The great advantage of IR is that you also have a very good database library for different compounds in IR, even better than NMR. Yeah, and even better than MSMS that GNPS will present. I think you have a wider database for IR. So which means you could use that as a dereplication. Regarding finding new compounds using IR, I'm not really sure because I'm not an IR expert. So maybe you need to combine IR with other spectroscopic methods like maybe MS or NMR just to find new compounds. But why not? I think I have one collaboration in Malaysia, UKM, where they're using also a lot of IR. But I think they do combine this with NMR to say, okay, I've got here a new compound. But if you get a new database, I just am not sure what's the name of the database of IR, but it is a large database. And if you could prepare a macro, you could do this by using Excel, where you could compare data points from a database and what you have. And if it will if it will not find the hit, then maybe that's the way of saying that you have the possibility of a new compound and targeting this new compound. Yeah, that's something new, Triana, that maybe you could develop. And if you could develop this methodology, it would be a great paper as well. Yeah, because as I've said, yeah, there is a very good IR database out there. But yes, but because I'm not an IR expert, yeah, I'm not really aware of it. But it's something that you could explore. Yeah.
Since IR will be somehow much more affordable in ours, because like LCM, SMS will be very expensive here, as well as the NMR. So in comparison between NMR and LCM, SMS, do you think which one better? Is that one it's better than the others, or it's better to combine, or how is it then?
In my opinion, yeah, it's better to combine, yeah, because then you have the strength of both analytical methods, okay, which means you are disregarding the disadvantage of both methods. Yeah, for example, well, I'm using MS and NMR because that's my expertise. But as I've said, there's no stopping using IR. I know the one group in Strathclyde as well, the fermentation group, they, for example, developed methods of using IR probe and putting into the big fermenters just to be able to, what do you call this, detect the production of a certain metabolite. Yeah, but they already know what IR spectra they expect. Yeah, and IR has been there for ages, for generations. That's why its database is also has been established. It's the same thing with GCMS. Yeah, electron impacts. So you've got a very good library of fragmentation in GCMS. I mean, it's uncomparable with what we have today because it has been there for generations. And the same thing with IR. I don't think anyone has just really explored it enough because everyone was using MS and NMR. I would if I'm an IR expert, or maybe I could still develop it. But yeah, if you're in front of an IR, you should develop it yourself. I mean, okay. I could help if you want to.
Yeah, of course. Love to.
Yeah.
And how about my second question, Ruan, about the honey chemical contents that you mentioned that it's somehow so that forgive me if I didn't pay attention enough. You mentioned before that the biological activity was different. The one from Malaysia was active towards the cytotoxic activity, but the other one from New Zealand were not active. But the NMR and MS, there was no difference. Yeah. But you mentioned something about the metabolomic. Can you maybe explain more?
Yes. So if you look at the NMR spectrum of Malaysian honey and New Zealand honey, there's no big difference because you're just seeing the big components, and that's a disadvantage of NMR because you could only see the major components really. And what's the major components of honey? Sugar, right? Fructose, glucose, so all the sugars. So that's what we see. Yeah. But then there should be a reason why one is active and inactive. So using a metabolomics approach and using MS, so you could still see the tinier peaks in the NMR that represent the minor components, but you need to be able to relate that of what we see in the mass spec. And the mass spec is the same thing, and we are only seeing lower molecular weight compounds, but using a metabolomics approach or using MZ-Mine where we could process our data, then we could put down the threshold and look at the smaller compounds that would make a difference between New Zealand and Tualang honey. And that's the way we were able to find out that they are indeed different. And these smaller or low yielding metabolites were important in the bioactivity of Malaysian honey. And that also explained the difference between with New Zealand honey and between North and South. Yeah, but we could only see that in the smaller peaks. And to do that manually is impossible. Yeah, so you need to really automate that system. And that's the reason, yeah, we're using macros, we're using MZ-Mine. So, and this is the importance of developing methodologies, right? And you could do this, definitely you could do this with IR as well. Yeah, if you just concentrate in the fingerprint region of the IR, then I think that will be able to define differences as well. I think I did one step with Malaysia. We had this lichen project, but I didn't know what happened after that. Or they were using IR, but I said, okay, let's concentrate the fingerprint region. And then you could really see the differences. Yeah.
Okay, thank you very much, Ruan.
You're very welcome, Djuhana.
Bye, Yasman.
Thank you, Dana. So, is there any question for our speaker? There's no additional chat. Pak Haidir, maybe, or Pak Edi?
Yasman's now calling roll calls. Are they still there?
Yeah.
Hi, Eddie. How are you?
Ruan.
Hi, Ruan.
Sada,
Glad to meet you.
Here's Dian.
I don't meet Pak Yasman in our lab in Wurzburg.
Oh, you don't know each other?
No.
I just know here in this Zoom link.
So Yasman started.
Düsseldorfer.
Yeah, Yasman is a Düsseldorfer.
Yeah. Okay, go on, Pak Edi.
No, I just a listener because my work noise outside from Metabolomic, and so that's let's say a reunion from our former colleagues and our former mentor during my stay in Düsseldorf. Thank you.
Yeah, you're very welcome. Well, I could just say generally, working with Metabolomics is you could find really a lot of application. And I think, when I started my work at Strathclyde, I really need to reinvent myself. I need to find something that people are not really doing. Yeah, so that's why I came into Metabolomics. And I just saw, okay, I could apply this to different things. And the same thing there, whatever instruments you have there. Yeah, as you said, having a mass spec and NMR is a large investment, but you could do collaboration. You could just take the results and then try to develop your own metabolomics method. That would be good. Or you could use other instrumentations as well. IR is being used, UV is being used. You could, I think one thing that I haven't seen in the literature is fusion of UV and IR. Those are things that you could explore. Yeah, why not? And if you could do that, then that could be a good paper. Yeah. Or GCMS, I think a lot of you has GCMS. The GCMS is not even high resolution data. But the nice thing working with GCMS is, I said, there's a very big supportive library for GCMS. Although it's not high resolution data, you could still do your replication with GCMS. So which means you could use that as well. Why not concatenate your fused data from GC and IR? Yeah, so there's really a lot of combinations possible to develop a new methodology. I came out with MS and NMR because that was my ex, and that's the instrument in front of me. But if I have an IR in front of me, I will develop IR and NMR or IR and GC. But I don't have an IR in front of me. So there's a lot of things to develop.
Okay, thank you for your insight. I try to find a way to put into the dictionary. Yeah, I hope now with GC and MS and also UV. So I think hopefully then I can find the approach to go to in the metabolomic direction. Then. Thank you for your insight.
Yeah, you just need a very good student who has a lot of patience to work with the computer and do programming. Then that would help a lot. Find a student who loves computer.
RuAn.
Yeah.
You just comparing the period where we're dealing with the natural product from the very conservative method, for instance, 5 to 6 years, but for the use of metabolomics method, we can shorter the period by 2 or 3 years.
Yeah.
My question is regarding to seeking a new natural product.
Yeah.
How faster we will get the new product with the use of metabolomic method compared to the classic or traditional method that I did in my Ph.D. project in this or your Ph.D. Because you know in metabolomic usually we use GCMS or LCMS, but then we seems to be trapped in working with the known compound. You know, we try to attenuate the peak with the library inside the GC or LCMS machine, and then we neglected the unknown compound or maybe the minor or something like that. It's therefore then I'm curious how fast you will get new natural product with metabolomic method compared to the traditional method.
I think you save a year, I guess. Yeah, because for example, so isolation.
But again, it is faster with using metabolic method than the
Yes, because then you don't look at every fraction. So the best comparison I could say is in the first fractionation stage, the student will usually get 22 fractions. But then only 3 fractions will be active. Then I would tell the student concentrate on the active fractions. And then you could revisit the inactive fractions if we could say related compounds, then we revisit that. But then in the active fractions, sometimes you would see that both active and inactive features are already there. And you could see that from the metabolomic maps or the metabolomic plots. Yeah. So yeah, so instead of working with 22 fractions, they just work with 3 or 4 fractions. I think I had one case, the latest one is she had 20 fractions, or he had 20 fractions. And how much is that? 12 fractions were active. Like, oh my God, 12 fractions. And we need to prioritize. Yeah. But then we could then look at the target metabolites and see the relative abundance of these target metabolites in each of the fractions, and each in the 12 fractions. And then she was able to go back to 3 fractions. Okay. And she's now on she started with me in November. Okay. And she's now doing the isolation work. So this particular project should finish by next month. Yeah. So I think it's quite fast, because then this could be a year work. Yeah. But then she finishes in 6 months. So it's really quick. Yeah. Because I remember when doing my PhD, I've got 12 fractions and I have to work out. So I need to judge my fractions according to, okay, bioactivity, right? And if half of them are active, I have to isolate. So each fractions will still contain maybe 10 compounds in it. But then I need to isolate each of the 10 compounds, right? Which is useless, because only of these 10 compounds, only 2 are actually important. Yeah, but with metabolomics, I already defined my target. Oh, this is my target compound. I don't need to isolate 10 compounds from each of the fractions. So remember during our PhD, how many vials do we accumulate?
Thousands.
Thousands, right? No, we don't do that anymore. They just fill up their fridge, and then at the end they have problems sorting things out.
Thank you, Ryan.
Okay.
Again, to the audience, is there any question?
No. Okay. Nice to see everyone. Kaidir, you're so quiet there.
Yeah. Pak Hadir.
Did I miss me?
Indomie Pak Hadir. Mie goreng.
Indomie.
He's the boss now, so the boss must remain silent.
I know. He's a big boss. He just listens and gives one.
Say something, Pak Hadir.
What? Oh yeah, I don't have any question for Ruan. I just happy to see you all in one screen. I hope we can meet with the others in.
You organize that.
I will organize that.
Okay, thank you very much.
And we will discuss about metabolomic more details.
Good. I keep your promise. I keep your promise, Pak Hadir.
Okay.
Thank you.
Okay.
So if there is no question anymore, there are actually 14 minutes more. But if this session is enough, I think we come to the end of this event. Again, I would like to thank Dr. RuAngelie Edrada-Ebel for a very nice lecture and very interesting and comprehensive discussion to all of us. I don't think that I have to wrap up all the lecture that given by RuAn, and also all the discussion that we have made. So we give a big applause to RuAn by reaction, of course, because it is online. Again, thank you very much, RuAn, and hope we will see in next or other event. And for all the participants, I would like also to thanks Pak Hadir, Bu Dian, Bu Triana, Bu Dewi. Who else that I forgot? Pak Edi, and also a lot of our students here, and also staff and lecturers in FMIPA UI. There's Professor Titin, Pak Heri Cahyana, and others that can join this event. Again, thank you very much for all. Stay healthy and always happy. Of course, I close this event for today. Again, thank you very much. Shall we again make a group of photo session? Pak Hadir, I don't know. He's still on cam or?
Yeah.
Yeah.
Okay. You're still there. You just woke up.
Thank you, Pak Dekan.
Halo Ari.
Halo Ibu. Halo Dr. RuAn.
Hai.
Halo.
Ya, Bu Ari from
Udayana.
Udayana ya, Unan.
The youngest apa ya?
The youngest cohort.
The youngest alumni from Prox ya.
Ya, foto-foto.
Mbak Yusin bisa ini bantu foto.
Okay, Pak. We have two screen and we start with the first screen. Okay, 1, 2, and 3, and the second one, 1, 2, and 3. Okay, thank you.
Okay, again, thank you very much, RuAn.
Thank you,
all of us.
Bye-bye, RuAn.
Thank you very much.
Bye,
RuAn.
Nice to meet you all.
You too.
Terima kasih Pak Yasman, Pak
Ya,
Mide,
sama-sama. Yuk.
Pak Hadir, Pak Dede, Pak Edi.